This video will be on the process of transcription. So transcription is carried out by RNA polymerase. In prokaryotes, RNA polymerase consists of two main parts: the core enzyme, which has this beta, beta prime, 2 alpha, and an omega subunit, and then the sigma factor as well. So once you have all of those, it's called the RNA polymerase holoenzyme. In eukaryotes, there are multiple kinds of RNA polymerases. There are three primary ones in all eukaryotes, and RNA polymerase II is the primary one that is used for messenger RNAs. So RNA polymerase I and III are used for particular types of functional RNAs that would be genes in which RNA is the final product of that gene. Also in plants, there are two other RNA polymerases. We're not going to go into those at all. So there are three stages of transcription: initiation, elongation, and termination. And these processes are very different in prokaryotes and eukaryotes. We're going to talk about all three of those processes, first of all in prokaryotes and then in eukaryotes. So how does the cell actually know where genes are in that long DNA sequence that is found on a chromosome? The way that it finds genes is actually via the promoter. So promoters have particular sequences that are in common among a lot of different genes. And scientists who have looked for where genes are have actually found something called consensus sequences in their promoters. So I just want to tell you, first of all, what a consensus sequence is. If you look at a lot of different promoters, right before the place where the gene starts being transcribed, you find certain sequences. And if you look at a lot of different genes, you'll find that there are some similarities between those sequences. What scientists can do, along with the aid of computers, is come up with a preferred sequence called a consensus sequence, which is the most likely base to be found in a particular position. So, for example, in this first position, most likely you're going to find a T. Second position would be an A. Third position would be any pyrimidine, and we actually indicate this with a Y. So it's either a C or a T. So prokaryotic promoters are recognized by particular sequences, and the place where transcription starts for a particular gene is always the +1 site. The promoter would come before that, so we call those minus bases. There are two places that are very important in prokaryotic promoters. Those are the -10 consensus sequence and the -35 consensus sequence. There's also another sequence that is frequently found. This is called the upstream element, and this would be found a little bit further before that start of that gene at around -40 to -60. So these consensus sequences at the -10 and -35 positions are recognized by the RNA polymerase sigma factor. And so sigma actually binds to those promoter sequences, and it will then recruit the rest of the RNA polymerase enzyme to attach to the DNA there and to begin transcription. What's actually going to happen then is, once the entire holoenzyme is bound at the promoter, the two strands of DNA are going to separate from each other, and it will position that RNA polymerase to start making the first RNA base right at the +1 site. So if we look at a summary slide here, the sigma factor is the actual factor that binds to the promoter, the -10 and -35 consensus sequences in prokaryotes. Then it's going to recruit the rest of the RNA polymerase enzyme, which is shown by this big pink oval. Once that entire holoenzyme is recruited to the promoter, then those two strands will get separated from each other, and the very first base will be added at the +1 site at the beginning of the gene. Once we get that first base added, then a couple of things happen. First of all, RNA polymerase will start moving along the DNA to the right. The sigma factor is actually going to be released from that holoenzyme. So the sigma factor is really only important in initiation of that transcription. It's not so important in the elongation itself. If we then continue to look at how elongation happens in prokaryotes, there is a particular region here that is going to remain single-stranded, and so the two strands of the DNA separate from each other, and that region, which is called the transcription bubble, is about 18 nucleotides in length. Approximately 8 nucleotides of that RNA are actually going to base pair with the DNA. So we make an RNA-DNA hybrid, and then as we continue making more RNA, which is going to continue on unwinding to the right here, we actually start to extrude that RNA that's being synthesized out of the transcription bubble. Then as the bubble continues to the right, we start closing up that bubble on the left side and rewinding the DNA together. The rate of transcription in prokaryotes is about 40 nucleotides per second. Once we continue on and get to the end of the gene, there are a number of things that have to happen for termination of transcription to occur. So the RNA polymerase has to stop transcribing, and this is not the end of the DNA template, right? The DNA will continue on. The RNA polymerase has to get some signal that it must stop. The RNA has to be freed from both the DNA and also from the RNA polymerase itself, and then RNA polymerase has to fall off of the DNA. So in prokaryotes, there are two different types of transcriptional terminators. There are, first of all, row-dependent and then row-independent terminators. Row-dependent terminators require a row protein. And what happens is that there is a particular sequence called the rut site, and that sequence will be transcribed and it will be in the RNA here, and row will actually bind to that rut site. When the row factor binds to the rut site, it actually causes the RNA that has just been transcribed to be formed into this conformation that is going to disrupt the interaction with the DNA. And so we form this complex, this hairpin, and it actually moves that row protein right into the transcription bubble, where it's going to actually disrupt the pairing between the DNA and RNA and make that RNA fall off of the DNA. At that point, the RNA polymerase as well will fall off and the DNA gets wound back up again. The second type of terminator is called row independent, and this is actually dependent only upon the sequence of the RNA that is transcribed. So what happens here is there's a region in the DNA that is called an inverted repeat. Now the DNA is double-stranded, so it's not going to make any unusual structures with itself. However, once that gets transcribed into RNA, we form this region of the RNA that can actually base pair with itself. This is called a hairpin loop, and this hairpin loop is followed by a string of U's. So remember, A's and T's in DNA and also A's and U's in the DNA RNA hybrid have only 2 hydrogen bonds between them, so it's a little bit weaker bond. And the presence of this hairpin loop along with this really weak binding causes things to get destabilized, and at this point that RNA will fall off of the transcription bubble, RNA polymerase falls off, and transcription is terminated. Let's now move on to eukaryotic transcription initiation. Let me just remind you that there are 3 RNA polymerases. Each of those RNA polymerases recognizes slightly different promoter sequences. So we are only going to talk about RNA polymerase II, which is the polymerase that synthesizes messenger RNAs. Another big difference in eukaryotes compared to prokaryotes is that eukaryotes have nucleosomes, and nucleosomes wrap the DNA, and later on we're going to discuss how this will actually affect transcription. There are a lot more factors that are required for transcription in eukaryotes than in prokaryotes, and so we're going to talk about some of these general transcription factors. We'll also talk about mediator. Transcription initiation in eukaryotes is immensely complicated. There are 2 main components to all eukaryotic promoters. There's the core promoter, and then there are regulatory elements. So the core promoter is right at the +1 site, and this is the place where RNA polymerase will actually bind. In addition to RNA polymerase, there are a whole bunch of other factors that need to bind to this core promoter, and altogether these are called the basal transcription apparatus. Now, the tricky thing here is that the core promoter all by itself is not sufficient to actually activate transcription at high levels in the cell. If you look at eukaryotic promoters, you're going to find a lot of diversity in these promoters that are all regulated by RNA polymerase II. There are 4 known consensus sequences, and most promoters have some combination of these 4. Not all promoters have to have all 4 of them, but they have to have some combination of them. So starting at the +1 site, at the transcription start site, right there is something called an initiator element. About 25 bases before that, some genes have a TATA box, which we'll talk about more in just a minute. And about 10 bases further upstream, we have the TF2B recognition element. Some genes also have a promoter element that is found after the start of transcription, and this would be called the downstream core promoter element at +30. Regulatory elements include the regulatory promoter, and this is found just before the core promoter on the DNA sequence. So here's the start of transcription, here is the core promoter, and before that would be found the regulatory promoter. And then they also have enhancers, and enhancers are found a lot further away from the core promoter, and they can actually be before the gene or after the gene in non-gene coding regions. The purpose of these regulatory elements is to help regulate both the activity of the core promoter and also the recruitment of the basal transcription apparatus to the core promoter. And it is the regulatory elements that provide for differential gene expression between different genes. If we look at what is required for eukaryotic transcription initiation, I just want to focus in on this region at the start of transcription. So this would be the +1 site, and the core promoter is located right before and around this transcription start site. Regulatory promoter would be up further before the gene. The things that you're going to need for the basal transcription apparatus are a bunch of general transcription factors, and these are all called TF2 something. So TF2 stands for transcription factor for RNA polymerase II, right? That's what the 2 is about. And their letters, so A, B, etc. There's another factor called mediator, and then of course RNA polymerase itself. So a lot of genes have a TATA box. So TATA is the consensus sequence, and this type of promoter has been studied pretty extensively, and we know that there is a factor called TATA binding protein, or TBP, that binds to that TATA box. Once TBP binds to the DNA, then it's going to recruit the general transcription factor TF2D. And so here TF2D binds, and once this gets bound, then it's going to help recruit a whole bunch of other factors, including these other general transcription factors, RNA polymerase, and also mediator into this complex. That core machinery is not going to be sufficient by itself. We are also going to need aspects of the regulatory elements in the promoter in order to activate transcription to high levels. In the regulatory promoter and in the enhancers, there are binding sites for what we call transcription factors. Transcription factors, shown in these purple circles, will bind to the DNA in this region, and they do a number of different things. First of all, they interact with the protein mediator to make a connection with that basal transcription apparatus. Sometimes they help to recruit the rest of the basal transcription apparatus, or they might act to move nucleosomes out of the way so that we can actually start transcription. When everything is bound, that whole basal transcription apparatus is ready to go. It's activated by these transcription factors that bind to the regulatory regions of the promoter. Then finally transcription can begin, and we'll separate those 2 strands of DNA right at this transcription start site, and the first base of RNA is made. One other point I'd just like to mention here is that The transcription factors that bind to the regulatory parts of the promoter, so the enhancer and the regulatory promoter, can also sometimes be repressive in nature. So sometimes those actually act to block or inhibit transcription of a gene. We will come back to that a little bit more when we talk about the regulation of gene expression. Eukaryotic elongation happens relatively similarly to prokaryotic transcription elongation. So first of all, when that RNA polymerase leaves the promoter, it's going to start synthesizing new RNA, and it's going to add new RNA bases on the 3' end of that RNA strand. When it leaves the promoter, many of the transcription factors, the general transcription factors, will remain at the promoter. So it's really only RNA polymerase that needs to do the transcription itself. A lot of that other stuff can actually stay sitting on the promoter and work to recruit another RNA polymerase so that we can transcribe that gene again. The transcription bubble is shown here. So here's the double-stranded DNA on either side, and here it is now opened up and used as a template to make the RNA. And this is very similar. There's about 8 bases of the RNA that base pair with the DNA, and the DNA then provides the template for making RNA. Once that RNA gets long enough, it begins to trail out of that transcription bubble, and the transcription bubble will then move along the DNA down the gene. Finally, transcription termination in eukaryotes is achieved based on a particular sequence. So when we get in the RNA to a particular sequence called the cleavage site, it will be bound by a protein which will then cut that RNA. And so it releases that pre-messenger RNA, which will then go on to finish being processed and be translated eventually. As far as RNA polymerase is concerned, RNA polymerase actually continues on. So the mechanism of termination does not directly affect the RNA polymerase. Sometimes you can continue on for several hundred bases. What happens to the RNA that is produced, however, is that it rapidly gets degraded. And so right here where this cleavage site was, a protein called RAT1 binds to that RNA and starts to chop it up. Pretty soon RAT1 will then catch up to the RNA polymerase, and once it degrades that entire RNA, then the RNA polymerase will at that point fall off of the DNA and transcription is terminated.